It gives me great pleasure to introduce today's plenary session, hosted by Dr. Carl June, Director of the Center of Cellular Immunotherapies, Perelman School of Medicine, and the Director of the Parker Institute for Cancer Immunotherapy at the University of Pennsylvania. Today, Carl June will be sharing the state of the art with CAR T therapy, and the title of his presentation is Interrogating the Genotype and Phenotype of CAR T cells in the tumor microenvironment.
Well, it gives me great pleasure to participate in this Advancing Science, the first COVID-induced virtual science program put on by NanoString. And today I'm going to talk about our results with CAR T cells and using the new GeoMx platform to interrogate the tumor microenvironment. My disclosures are here. The relevant ones are Novartis and Immunotherapies. So about 20 years ago, we began working on CAR T cells, initially in HIV infection, and then later in patients with various cancers. And the CARs became more and more sophisticated over time with the incorporation of co-stimulatory domains. The initial CAR had only the zeta chain of the T cell receptor, and that led to limited proliferation of the CAR T cells when they found antigen, but they did persist very well. In fact, we have data that they are still in patients more than 15 years since their first infusion. However, those first generation CAR cells, when they were tried in cancer, did not work. In that they persisted for only a few days in the cancer patients, probably related to the more toxic immune environments in cancer patients than they are in patients who have HIV and who are on antiretroviral therapies. So the field then began to test CAR T cells having more complex co-stimulatory domains with so-called hardwiring of the co-stimulation, such as CD28 or 4-1BB, into the CAR T cell. And those began trials in 2010 and 11, and then became FDA approved in 2017, with Yescarta having a CD28 signaling domain, now marketed by Gilead, and Kymriah having a 4-1BB signaling domain and now marketed by Novartis for leukemia and lymphoma. And so that began a new cell therapy industry in that this is very different than the standard pharmaceutical model where the drug company will make a small molecule and in large lots and distribute it to many thousands of patients. In this case, the CAR T cells that are initially FDA approved are in so-called N of 1 cell products, in that they're autologous and derived from the patient, and then manufactured, and then cryopreserved and fused into the patient. So there are many steps on this, but the logistics much more complex, certainly, than standard pharmaceutical approaches. And we're still in the midst of this new industry in making the cell therapy and manufacturing more automated and less labor complex. But in the case of leukemia, there was no real issue with studying the CAR T cells in the patient because they circulate in the patient and also are easily found during standard of care bone marrow biopsies. However, with solid tumors, the CAR T cells leave the blood and go into the tumor microenvironment, and that's what I'll be focusing on today. So there are basic differences between these two commercially marketed CAR T cells, both in their vector design, from lentiviral vectors or retroviral vectors, the promoters, and they both have the same binder to the CD19 molecule that's found on normal B cells and leukemia cells or lymphoma cells. They have different co-stimulatory domains, as I pointed out a moment ago, different hinge and transmembrane domains, and very different persistence in patients. And we believe most of that is difference in persistence in patients is due to the differences in the co-stimulatory domains, CD28, compared to 4-1BB. My colleagues, David Porter and Jos Mellenhorst and Joe Frietta, have studied now the first 2 patients we treated. In this paper published in 2018, they showed that the CAR T cells had survived for more than 5 years in both patients. And we think these are actually not the initial cells infused into the patient, but the progeny or daughter cells of those who divided in the patient and yet continue to carry the transgene for the CAR because they have an integration, generally 1 or 2 copies of the CAR per T cell. So this really is what's transformational for the pharmaceutical industry. It's the demonstration that you can create a living drug that will persist in patients for years. And we showed in that work that we could re-isolate the cells from patients and that they would then still be able to kill leukemia cells in a CD19 specific manner. So they remain on immunosurveillance even after the patient has been induced into remission. And we studied both in acute leukemia patients and chronic lymphocytic leukemia patients, the long-term surviving patients and the CAR T cells that we could re-isolate from their blood. And in this slide in panel A is, on the left, is demonstration of the CAR survival measuring both by a PCR assay, the number of copies of the CAR T cell transgene in blood on a log scale. And you can see there's about a 2 log difference in a patient who had a complete response compared to those who had non-response or NR. And below that on the left is a plot of the normal and malignant B cells in the patient. And you can see that the patients with complete remission have the levels of B cells become undetectable below the limit of detection in the assays, whereas the non-responding and partial responding patients have either no response or a partial response. So this is our first clue that the proliferation of the CAR T cells in patients is associated with responses. And we've since found that in many other kinds of cancer beyond CLL, for instance, in lymphomas and in patients with myeloma. So on panel B on the right, we studied some of these long-term persisting CAR T cells, and in this case, cells in the infusion product in the patient. And what we find is that there's a very large difference in, for instance, in panel D, the amount of memory cells in the infused product, and that's higher in the patients with CR or complete remission than in the partial or non-responding patients. Conversely, in the lower right in panel G, we find that if you measure genes signatures associated with T cell exhaustion, that those are very low in the infused product in responding patients, and exhaustion is easily detectable in the non-responding cohort. So the quality of the infused cells is important in determining the later response of the patient. We continued our initial studies in acute leukemia and had, as a swimmer plot shows, the patients with complete responses, there was an extraordinarily high complete response rate in patients with relapsed and refractory leukemia, with over 90% of the patients in remission within a month after treatment. So in a phase 1 trial of refractory and relapsed patients, this is the highest response rate that had ever been recorded. These data and others then replicated at Novartis led to FDA approval in August of 2007 for pediatric and young adult leukemia ages 3 to 25. So this was the first time that a cell and gene therapy had been and had reached commercial approval in the United States by the Food and Drug Administration. But what I'll do now is turn to CAR T-cell therapy for solid tumors. And the elephant in the room for that is really that many trials are underway in patients with solid tumors, but as of yet, there have been no spectacular responses, as many groups have now seen for hematologic or blood cancer. So there are many reasons probably why solid tumors are harder to treat with engineered T cells. And those could go from trafficking into the solid tumor to the more toxic tumor microenvironment. And so the central challenge to the entire field of cell therapy now is to identify those barriers so that they can be engineered around in order to improve the efficacy of CAR T cells in patients with solid tumors. And in the next several slides, I'll focus initially with our CAR T cell trials targeting mesothelin in pancreatic and ovarian cancer, and then at the end turn to prostate cancer as a model of a solid tumor treated with engineered cells. So there are many reasons as potentially as to why the efficacy is diminished with solid tumor therapy compared to bone marrow-derived malignancies. This cartoon shows that there are intrinsic cell qualities that may change in the solid tumor panel on the patients on the left. That the tumor is more likely to have the T cells have poor quality in the patient, even when they're taken out of the patient for manufacturing. So we have recent and unpublished studies in pancreatic cancer showing that the phenotype of circulating cells, when one looks at naive cells and effector memory and central memory cells with high dimensional time of flight flow cytometry, that they're perturbed in untreated patients with pancreatic cancer compared to age-matched controls. So that is one area. On the right panel is a cartoon depicting the tumor microenvironment in a solid tumor, and there are many barriers there, from collagen that may be limiting access by diffusion and cell trafficking into the tumor to many more suppressive cells, such as tumor-derived Tregs or myeloid-derived suppressor cells and tumor-associated macrophages. So these all contribute to a more complex and toxic tumor microenvironment, and include also the metabolic perturbations such as acidosis and hypoxia in many solid tumor microenvironments. So we've been working with NanoString now for almost 2 years, trying to understand how we could study these complexities in the tumor microenvironments with CAR T cells. And we first began to test in mouse models with NSG mice that are xenografted with human CAR T cells. And then we have moved more recently now to studying patients on our mesothelin trials with pancreatic and ovarian cancer, and then are just beginning studies in the trial of prostate cancer. Pancreatic cancer is one of the worst cancers as far as prognosis, because at the time of diagnosis, it is usually unresectable. One target in pancreatic cancer that a CAR T cell could access is mesothelin. This is a GPI-linked protein that's overexpressed on more than 90% of pancreatic cancer cells, as well as some other solid tumors such as ovarian cancer and mesothelioma. So we have made several CAR T cells now specific for mesothelin over the past 15 years. The first one we did in collaboration with Ira Paston at the National Cancer Institute, and he had made an antibody called SS1 that bound to the N-terminus of mesothelin. Later, in collaboration with Novartis, we made a CAR against a more membrane proximal epitope, and this CAR is derived from human antibody sequences as compared to the SS1 that I mentioned, which is derived from a mouse hybridoma. As you can see on the top left panel on BioCore analysis, there is quite a large difference in the KD, or dissociation constant, of the SS1 binding to immobilized mesothelin compared to the M5. So the M5 CAR has about a 30-fold lower affinity and KD for mesothelin than SS1. In addition, as shown on the bottom left, the mapping of the epitope showed that the SS1 CAR binds to the N-terminus of mesothelin, and that the M5 CAR binds to a membrane proximal epitope. We then made these in the same format with CAR construction as with Kymriah, so that they have a a 4-1BB signaling domain, as well as a zeta chain expressed in T cells using a lentiviral vector. So very similar to how we do the manufacturing in leukemia, but just swapping out the CAR design. So we now have done a series of clinical trials, phase 1 and pilot trials, testing these CARs in patients with pancreatic cancer and ovarian and mesothelioma. So the initial trials were testing SS1, and these studies are published. I'll briefly summarize them. And we have ongoing trials with the M5 human-based antibody CAR that are open now for both mesothelioma, ovarian, and lung cancer. And you can find details about these trials with their NCT numbers at clinicaltrials.gov. And this paper, published in 2018, summarized our second trial in metastatic pancreatic ductal adenocarcinoma, or PDA. And we had 6 patients in that trial. And in panel C on the left, the patient was measured at baseline and then after a month after treatment. And the PET scanning with FDG PET is used to assess tumor activity. And 1 out of the 6 patients had a partial response, as you can see, patient 113 in the green bar that goes down. The other patients had either stable disease or progressive disease. In panel E in the center is shown that patient 113 with, on PET scanning at baseline, a very active and multiple metastatic lesions in the liver. And at a month after treatment, those are no longer PET avid, which is also seen in the unenhanced CT images and the PET CT combined images shown in panel F on the right and the bottom, so that the patient's disease regressed after a single infusion of these CAR T cells. And the next slide here shows that there's some evidence for metastatic, epitope spreading. And on the top, on panel B in these 6 patients, we plotted cytokines in the patients and show that for 5 out of the 6 patients, the cytokines in the serum didn't change in the 30 days after infusion, whereas patient 113, who experienced an anti-tumor response, is the only one who had elevations in cytokines, particularly IL-6, IL-1, and IL-18. We have also seen with our CD19 CARs that elevations in cytokines, such as these shown here, correlate with response in the patients. We found that The patients made new antibodies against neoantigens using the protoarray in panel C. So this was some evidence that there had been new humoral responses made in the patients against a variety of recombinant proteins. And in panel D has shown that the CAR T cells actually infiltrated metastatic lesions that could be tested at biopsy time, biopsies post-infusion. So those trials enabled us then to move to testing the fully human CAR called M5. And data I didn't show you, the CAR T cells did not persist long in that SS1 CAR trial that I just reviewed. And what we found was that the patients made immune responses against the CAR molecule, probably because it was derived from a mouse hybridoma and is immunogenic in humans. So now we're testing this M5 CAR that has a lower affinity and also is fully human. It turns out it's much more potent in two different cancer models on the left. In studies done at Novartis, they tested infusions of the M5 CAR or infusions of the SS1 mouse-based CAR against a xenograft in immunodeficient mice, and only the M5 CAR was able to cure those mice in data out to 63 days after implantation of the tumor. And on the right is a series of pancreatic PDX models. So these are pancreatic cancers from single patients implanted into mice. And on the black is control T cells for allogeneic or xenogenic effects in these mice. And CAR-19 was given as a control since it targets CD19, which is not present on pancreatic cancer. And then the M5 CAR. And as you can see, almost all the mice treated with the M5 CAR had tumor regression in this very rigorous model. So in a head-to-head comparison, the M5 CAR is more potent in our preclinical studies than the SS1 CAR. And that trial is underway now in two different trials with that M5 CAR. And so we have pancreatic tumor biopsy specimens from those patients, as well as a variety of other samples, such as ascites and so on now, to study the activity of the CAR and then how the tumor might respond to the CAR targeting. So but what I showed you is there are some responses, but no one has had a complete remission to date with the CAR T cells against pancreatic cancer that we have tested. And this is where now we are working with NanoString to interrogate the tumor microenvironment and ask why we see some responses in some patients and no responses in others. So the GeoMx Digital Spatial Profiler is an instrument that's quite complex, but in basic, it takes an FFPE specimen from a biopsy specimen of a mouse or a human tumor specimen. Then regions of interest, or ROI, are selected using the computer and a microscope-based approach to examine in detail the protein and RNA content of those regions of interest. Then the antibodies and RNA hybridizations are cleaved off with UV light so that they can then be, with a microcapillary tip, aspirated off and then analyzed for the protein counting, which would be the same as IHC, and then also RNA to get an idea of the transcriptomics of the tumor microenvironment and the CAR cells. So initially, the samples I'll show you have been run through the NanoString M counter to get the readout, and there are many different codes within each transcript so that only those that have all exon tags as identified are registered as a true signal. Later, NanoString will come out with a NGS or next generation sequencing approach using, for instance, an Illumina platform, as well as the nCounter readout. They expect this more broad application to be probably come out, I think, in the third quarter of this year, although that may be in flux given COVID uncertainties. So we first began to analyze samples of mice with human tumor xenografts treated with human T cells. And this is work with a graduate student in my lab, Sonia Argawal, and a postdoc, Maria Aznar Gomez. And so the basic design of these experiments is that immunodeficient NSG mice were injected either with an ovarian cancer line called SKOV3 that has a lot of overexpression of the HER2 target. And in addition, the pancreatic cancer line, ASPC1. And then the mice were treated with either a Herceptin-based CAR or with an anti-mesothelin CAR, or UTD T cells, which is untransduced T cells. And then the tumors were, after mice were sacrificed, tumors were removed and then processed through FFPE and analyzed by the DSP system. On the right panel shows that the SKOV3 tumors do completely regress in mice treated with, in this example, an anti-mesothelin CAR T cell. And so we've used that model as a way to judge resistance mechanisms and how to improve CAR T cell designs. So our first experiment then was to test in the explanted tumor models what and identify regions of interest, ROI. So one of the most important aspects that we found with this application of GeoMx DSP technology to CAR T cells is to really understand how and where to set the regions of interest. Depends on what your question is. For instance, do you want to know if the tumor still expresses the target, in this case, Herceptin, or the Herceptin target HER2? Are CAR T cells present in the tumor, et cetera? So in this tumor experiment, we were able to use 3 different colors really, to understand whether T cells were present using CD3 staining in blue, HER2, the tumor surrogate antigen, which is in red, and DNA staining, which is in blue. And you can see in some of these regions of interest that some are almost devoid of T cells, and others have concentrations such as in ROI2. There are areas of many CAR T cells and others less. And then there are also tumors that are bright for HER2 and others that are less. However, what we realized in this experiment was we were unable to discriminate between CAR T cells and bystander T cells. This is a huge potential advantage of this technology, to understand the infused T cells in their state compared to cells that may have already been infiltrating the tumor, the so-called TIL, that are not necessarily antigen specific and certainly don't have the CAR molecule. So to do that, we have combined RNA-scope technology with the GeoMx DSP platform. And so this is a collaboration between ACD and NanoString. And this approach now, the pipeline's modified in that we first identify where the CAR T cells are by using RNA-scope and in situ hybridization to identify a sequence in the CAR molecule. In this case, it's in the 3' untranslated region. Then once we know where CAR T cells are, we can design in the regions of interest to find areas that are hot in that they have a lot of CAR T cells in the area, or others that are devoid of T cells, and others that are tumor rich or tumor poor. So the downstream processing is similar to the standard GeoMx DSP platform. It's the combination up front of RNA-scope that allows us now to understand the difference between a CD3 positive T cell, which could be a bystander T cell or a transgenic CAR T cell. So that would stain with CD3 in both cases versus the CAR T cell that will only be bound and show RNA scope hybridization through the hybrid for the 3' untranslated region of the CAR molecule. So this has proved to be quite exciting. In this slide, on the right panel is a xenograft tumor that was excised from a mouse. This is the ASPC1 pancreatic model, and the mouse was treated with human T cells expressing the CM5 anti-mesothelin CAR. And as you can see, some areas are deep, just green, and have no other markers. So those are basically pure tumor regions. Other regions are the pink and yellow, and in some cases they're merged together. So those are T cells that are CD3 positive. So you can see that there are T cell rich areas in this tumor and others that are T cell devoid. And on the left side, on the upper left of the 4 panels, you can see some cells that are clearly yellow, and others that are pink, and others that are green. So there are, in this case, 3 different markers used to inform where to select our regions of interest. So the next question then became, and this is, what size should our region of interest be? We began with ROIs that were circles 200 microns in diameter. And you can see in this tumor set of that there were ROIs identified on the upper left. There, some are completely green. Some have many yellow areas in there, and pink areas where they're very highly enriched for CAR T cells. And then others on the lower right where there's lots of tumor and also T cells and CD3 staining. So this was then subjected to various analyses. And these are the results with the ROIs that we showed before, set at 200 microns. And what you can see here on the top left is the genes, canonical gene CD3 epsilon. And these samples then were processed, used either a manual processing before loading into the GeoMx instrument, or using the Leica tissue processor that's automatic. And you can see there's a very high correlation in the T cell genes, CD3, the WPRE CAR gene, as well as on the lower left, the mesothelin gene, which is in the tumor cells in those images. And on this slide is the RNA results from that experiment looking at the RNA. So this is an 84-plex, and we were able to custom spike in some extra genes to the standard package of 84 probes. And one of the ones we spiked in was the WPRE gene, which is, as I mentioned, 3' untranslated region in the CAR construct. And here the ROIs are segmented out into CAR T cells only, and CAR T cells mixed in with tumor, and finally at the bottom on the right, you can see tumor only. And it's quite interesting now that we can identify a CAR T cell and separate that from the rest. So you can see in the tumor only area, many genes that are in the center that are blue, on the bottom, tumor only, are genes that are T cell genes. And the arrows highlight CD3 epsilon, and you can see CAR T cells only on the top are bright red on the heat scale. And similarly, WPRE. So we have now specifically identified CAR T cells in an FFPE embedded flank tumor specimen from a mouse that had a pancreatic tumor xenograft. So this has been a goal for a long time now, to be able to look at the genetics, both at the transcriptional level and by IHC of cells interacting with the tumor microenvironment. And this first example here in a mouse xenograft looks very promising. So other than choosing where to set the size, the location of the ROI, as we discussed previously, in this experiment, we asked what the size of the ROI could be. Could we decrease it from 200 microns down to nearly single cell resolution at 10 microns? And so what we've done here is set ROIs along that, as you can see on the right, from 200 to 10, and then looked at the number of RNA probes that were detected. And you can see that it's quite stable, plateaued down to about an ROI of 50, and then after that, the sensitivity falls off. So it looks like certainly using an ROI of 100 at the current level of detection of these probes should be quite sensitive for detecting transcripts in those fields. So next, I'd like to turn to where we're going to go next with this project, which is to analyze the clinical trial specimens from a trial we have in prostate cancer. And this is a CAR T cell that targets PSMA, the prostate-specific membrane antigen that carries on. It's an extracellular ectoenzyme involved in metabolizing glutamate and folate. And then PSMA has been known to be on prostate adenocarcinoma and also to go up across various ethnic groups with tumors of advancing tumor grade and stage. In this CAR we've made, it's so-called an armored CAR, in that we've put in a dominant negative receptor to TGF-beta. And the rationale for that is that TGF-beta is secreted by prostate cancer cells, and it promotes the growth and establishment of the tumor stroma, and as well has a very potent immunosuppressive assay on T cells that's been well characterized over the years. So Chris Kloss, when he was in my lab, designed the PSMA dominant negative TGF-beta armored CAR. And the Lanaval vector is shown on the left. It has the single chain FV to PSMA, then coupled with T2A system to the dominant negative TGF-beta receptor. And he tested that in mouse models and showed that it was efficacious. And most strikingly, and not something we actually expected to find, was he did a stress test with a prostate cancer line called PC3 that expresses the CAR target PSMA. And on the lower right, you can see that the growth of the CAR T cells in the culture when irradiated cancer cells were added at weekly intervals to the culture. And if the CAR T cells had the dominant negative receptor, they proliferated, and they proliferated for about 3 weeks longer than if they just had the CAR alone, which is in green and denoted by PBBZ. So that the dominant negative receptor both makes them insensitive to TGF-beta, and what we showed in this paper is makes them resistant to exhaustion, which at this point the field thinks is a major issue with the efficacy and holding back the efficacy of CAR T cells. So we were able to take those preclinical studies and begin a clinical trial, a phase 1 trial led by Naomi Haast and Vivek Narayan. The trial design is shown here, where the first 2 cohorts received CAR T cells, and then without any lymphodepleting, and then the third cohort has mild lymphodepletion with Cytoxan. And this trial is open at the University of Pennsylvania and now at several other centers around the country. And what we found is in those initial two cohorts was that the CAR T cells engrafted and proliferated, as shown on the right, with the same peak, the time to peak kinetics, or the Cmax, as we've seen in our leukemia patients. But here they're about 2 logs lower in number. If you look at the CAR copies per microgram of genomic DNA, it's around at peak in 2 out of these 3 patients at 2,000 to 3,000 copies per microgram of genomic DNA. And so that's about 2 logs less than what we've seen in our leukemia studies. But we're excited by the first patient to go in cohort 3, who's shown here. So this patient got the same dose of 1 to 3 times 10 to the 8th CARs per meter squared, but in addition had the dose of Cytoxan given the week prior to CAR T cell infusion. And shown on this graph, which is not linear, is that there was an exponential increase in PSA in this patient on the previous therapies he had that were failing. And then the CAR T cells were infused on day 0. And you can see that there was a very rapid drop to 0 within 3 weeks after infusing with CAR T cells of PSA. It became undetectable. He then had a grade 4 or 3, grade 4 cytokine release syndrome and macrophage activation syndrome that we've seen in our leukemia patients that are responding with very high levels of ferritin, as you can see in the green plot. So he had cytokine release syndrome as toxicity and an anti-tumor effect measured both on circulating tumor cells and PSA. So this protocol looks promising, but of course there is toxicity. And I think the best way to understand what's happening is to analyze tissue biopsies and apply those to the GeoMx DSP platform. And Joe Frietta and Naomi Haas are working with NanoString to do this, and then to interrogate these samples using the Cancer Transcriptome Atlas that NanoString has put together, where there's about 2,000 genes that their pathways can be profiled in biopsy specimens. So our approach here is to test the baseline biopsy, and then one that is obtained about 10 days after the patient's treated. And then, so looking at the tumor and baseline, and then after CAR treatment to examine, for instance, whether or not the PSMA expression has been modulated in any way. In addition, we want to compare the transcriptomic state of the CAR T cells in the biopsy specimen to the infusion product that we retain before from the infusion on day 0. So for that, we're going to have to do a cross-platform set of experiments to learn how to compare the NanoString and counter results from the infused cells to what we get on the GeoMx DSP platform. So this is a project underway, and we don't yet have results to show. So I'll just finish with 2 slides showing where we're also going to apply that, which is we've recently published in Science the results of using multiplex human genome engineering using CRISPR-Cas9 to remove negative checkpoints in T cells, the PD-1 gene, as well as the T cell receptor alpha and beta genes, and insert a transgenic T cell receptor. And the results of those studies were published in February, and a summary of those 3 patients showed that it was safe. We had no off-target effects that were unanticipated. The best response was stable disease in 2 out of 3 patients. And we had persistence of these gene-modified T cells and gene-edited cells. Shown on the right in patient 07 was that at about 30 days after infusion, about 10% of those cells had T cell receptor disruption as well as PD-1 disruption. So, and we showed with single cell analyses that in fact this was at the level of the single human genome. We were able to obtain editing of up to 3 genes at one time. So our long-term goal then would be able to apply this DSP platform to those kinds of experiments where we've used genetic modification of the cells as well as genetic editing. And geomics is going to be improved for that because NanoString is working on incorporating next generation sequencing to these samples. As I mentioned earlier, at this point, we're using nCounter, but with next generation sequencing, we should be able to broaden out what we do and incorporate even, I think, eventually some of the uses endpoints are modified cells that we've done using CRISPR-Cas9 technology and be able to quantify gene edits would be a long-term goal using this technology. So in addition to applying the Cancer Transcriptome Atlas, there's a number of other things we'll be able to do, which when we get down to a single cell level of resolution in understanding the tumor microenvironment. And I think, one thing is that the engineered T cells will have a real advantage over checkpoints, analyzing resistance to checkpoint therapies, because the engineered T cells can actually be identified, as we showed, for instance, with the WPRE analysis in this kind of assay. So we can determine whether it's a bystander cell or whether it's the engineered T cell we were hoping to test its therapeutic efficacy. So I see a bright future ahead in applying this to our studies. And in summary, just say that the DSP technology now allows us to have high-dimensional RNA and protein profiling of tumor biopsies in our mouse preclinical experiments, as well as in clinical trial specimens and the ongoing collaboration between the University of Pennsylvania and NanoString. We've shown that this technology allows us to identify CAR T cells in the tumor microenvironment, identify the tumor, and look at both levels of protein expression that are selected, and as well as the transcripted state of those T cells. So in the long run, it will be, I think, a very powerful technology to understand the immune health of the cells that we have infused, and we want to understand if they're healthy effector cells, memory cells, or are they tired and exhausted T cells, for instance, which is unfortunately what the tumor is very good at inducing. And I'll conclude. I would like to thank all the people in my lab. Joe Frietta is on the extreme left, and who's the one who has done the work that I showed recently on the prostate cancer and also on the CRISPR-Cas9 editing paper that was in Science. And many colleagues at Penn to thank for this, for these data, and they're shown here at the Center for Cellular Immunotherapies. Bruce Levine and Don Siegel, who've done all the manufacturing in the cell and vaccine and production facility, and the prostate cancer team and the pancreatic M5 clinical team, and as well as our colleagues at NanoString, Sarah Church, Isabel Lee, Christina Bailey, and Nero Ramachandran. So I'd like to thank you very much for your attention, and then I think we'll be able to take some questions.
It would help if I was unmuted. Thank you, Carl June, for that presentation. We're on behalf of our partners, ACD, Carl, and then on behalf of NanoString as well, we're thrilled to be working with you, especially to be able to tease out the difference between bystander T cells and engineered CAR T cells, and try to be able to tease out the biology between that and the interaction. So thrilled to be working with you on this. We have a series of questions for you. Let me maybe start by first just on the disease type. I know in a lot of the presentations, Carl, you were talking about pancreatic and maybe doing some work with prostate cancer. In terms of as you enter into solid tumors with CAR T, are there some solid tumors that you think are more susceptible to be treated by CAR T than others? And how do you think about which solid tumor you go after first?
So that's a great question, Nero. When we look at tumors from surgical biopsy specimens and test them in basically petri dish experiments, we find that almost all cancer cells can be killed by T cells if we have a CAR target that we do and test, so that if the CAR T cells can get there in a sufficient number and overcome resistance mechanisms and so on, I think all cancer cells can be targeted in principle. So that there are many issues such as fibrous capsules around some tumors that prevent ingress of cells. So then another strategy might be needed. There are many checkpoints that differ between different tumors. That's an emerging principle that some tumors rely mainly on defense by, for instance, a lot of regulatory T cells. Other tumors use myeloid cells, such as myeloid-derived suppressor cells or alternatively activated macrophages, to prevent the T cells from doing their job. So, this is a very active area of research now. And I think, no one suspected 5 years ago that lung cancer, which no one thought of as an immunogenic tumor, that now the front therapy for lung cancer is immunotherapy.
That's really true. Now, the question from the audience is around pancreatic cancer and why the prognosis for pancreatic cancer is especially so bad.
Well, that's a really good question. Pancreatic cancer is almost always has a mutation in the KRAS gene, which makes that active. And one aspect is that pancreatic cancer really is not detected early, so that it's possible that if we had a test like a pap smear for cervical cancer, that early detection would change this problem with pancreatic cancer, where, as I mentioned now, it's usually only detected when it's too late. So early detection would matter. And then pancreatic cancer has its own special immune defenses. One of those is that there are very few T cells actually in a pancreatic cancer. So it's very different than, for instance, lung cancer, where oftentimes at surgery you find many T cells already in the tumor specimen, and there's an ongoing attempt by the immune system to reject the tumor. In pancreatic cancer, it's like the immune system basically doesn't even know it's there. That's called immune ignorance in immunology terms.
Interesting. Okay. Then, Carl, I know you talked a little bit about how you're thinking about using DSP, and you showed one of the data pieces on going down a region of interest size, small features. Can you maybe comment a little bit about how the technology might achieve, single cell resolution and what the turnover time of the assay might be?
That's a great question. Getting down to single cell resolution would be an amazing advance. Already right now, it's such the ability to discriminate cells that you've engineered from the passenger cells that are already there is going to be a big advance. And so I think, at this point, the turnover time is, when you have experience, you can do 8 to 16 slides in a day and a half, 2.5 days. This is much better than, say, mass cytometry-based assays, which are just that to scale out to clinical trials.
Got it. And then I know most of your experiments have been with tissue samples. I'm curious about like bone marrow samples and things like that. Would you ask, first using the sample type, but also would you ask different questions in those sample types than you would do in sort of a solid tumor tissue?
Oh yeah, there's bone marrow. So there are a couple issues. We have either aspirates and biopsies, and in general, when they process a bone marrow biopsy, they decalcify it, and for the bone spicules, and that then can make technical problems with immunoassessing. So from what I'm understanding, it's going to be possible to do bone marrow specimens, and I think this will be a huge advance. We don't really understand why in leukemia, for instance, there are two main leukemias. One's the lymphoid leukemia, and the immune response is very different than it is in myeloid leukemia. And it seems like their stem cell niche is different, and so this technology, I think, is going to be very useful to uncover the basic immunobiology of stem cells and then their leukemic counterparts.
Got it. And Carl, one last question I have for you. The whole use of CRISPR-Cas9 is an interesting twist and capability you have in terms of how you might be able to modulate tissue samples. Can you maybe elaborate on how CRISPR is going to be adopted going forward into CAR T therapy in terms of how people might start using CRISPR going forward?
Yeah, so, this is one of these great lessons of basic science and how disparate fields can come together. I think when, genomic editing tools first evolved, people are looking at correcting in vivo gene defects, such as, for instance, thalassemia and so on. And it's turned out now that with CRISPR, it is so flexible that multiple genes can be targeted at one time. So literally, the possibilities are really amazing. To ability to improve immune cells so that they're resistant to the normal issues that a tumor will do to turn them off. So I think that so that's one possibility, which is it will extend activity in solid tumors. The other is this emerging whole new field of making other cells that aren't from the patient be CAR T cells. So using, for instance, cord blood cells that are normally thrown away when a baby's born, CRISPR can make those cells now to be used in other individuals by engineering out molecules such as a T cell receptor. So we're going to see many applications of genome engineering to cell therapy.
Got it. Carl, there's a question here around what's the risk of having overactivation, cytokine storms with knockouts of inhibitory receptors via gene editing?
Yeah, well, that's a really good question. And, when you turn off the immune system checkpoints, the main risk is going to be autoimmune disease. And so, we need more time to understand how often that will happen. We do know with checkpoint therapy, when antibodies are given to block checkpoints, about 20% of people have autoimmune side effects, the most common being thyroid dysfunction, hypothyroidism that can occur in addition to colitis. So far with T cell therapies that have been CAR T cells, the incidence has been lower. But cytokine storm could happen if, many T cells see antigen at one time. So, and those really at this point are hard to predict in animal models. This is one area where a human clinical trial is best to figure out safety in these kinds of settings.
Well, Carl, I know we're up on the top of the hour. I want to thank you so much for the content and taking questions this morning. And to all the audience, thank you for attending. I hope you enjoy the rest of the conference.
Well, thank you very much, Nero. It's been my pleasure.
Thanks so much, Carl. Stay safe and enjoy the rest of the day.
You too. Bye-bye.
Bye-bye.